Hi everyone, my name is Love, and today I'll be going over Dr. Maduro's practice midterm. To jump right in, I'll start off with number one, and it's asking which of the molecules shown here is an amino acid. So basically, to start off with, an amino acid has a backbone structure of NCC, right? So it'd have an amino group, a carbon, and a carboxylic acid group over here. This middle group is where your side chain will be. So we'll label that R, and another thing connected to it would be a hydrogen. So this is the general structure we're going to be looking for when we're trying to find amino acid. So let's look at the molecules on the right. So if we look at molecule 1, we see that we have an N here, a C here, and a carboxylic acid group here. So it meets our NCC structure pretty well. And our side chain is an H, and in this case, it's an amino acid, and in fact, it's going to be glycine. If we look at number 2, we see that we have an N, an amino group. However, our next thing that we're connected to is a carboxylic acid group. This does not meet the requirements of NCC, therefore it won't count. 3, we see that we have an amino group over here. We have another carbon here, and then we have a carboxylic acid group at the end. This follows the NCC structure with the side chain group right here, and this is an amino acid. So in this case, we can see that our amino acids are molecules 1 and 3, which our answer would be D. So moving on to our next problem, we can see that it's asking a strand of RNA has a sequence of 5' AAACG3'. It would probably pair up with an RNA strand sequence of blank. So first thing to consider is that we're talking about RNA over here. So our answer choice should have uracils instead of thymine, so we can cancel out any answer choice that has thymine. So answer choice C and answer choice D would automatically be canceled out. Next, we'd want to match up a complementary sequence to what we have here. So you know that DNA is anti-parallel. So if this is 5', then its opposite end would be 3'. Since this is 3', this end would be 5'. And would fill in the complementary sequence. So since this is adenine, in RNA, the complement would be uracil. So U, 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 C goes to G, and G goes to C. So as we can see that this would be our answer choice. So we're looking for some sort of variation of this. So if we look at E, we see a 5' U, U, U. In this case, we have a 5' C, G, U. So we can cancel out E. B says 5' GCA. We have 5' CGU. Still doesn't match up. And finally, A has 5' CGU, and our 5' CGU matches up. And if you keep going, you can see that the UU 3' comes in at the end as well. So our answer in this case is A. So moving on, this question is asking the bonds joining together individual nucleotides to one another in one strand of RNA are called. So if we're looking at a single strand of RNA and its nucleotides, well, we know that it's made up of a sugar phosphate backbone, right? And one nucleotide contains a sugar, phosphate, and a nitrogenous base, right? And this is what makes up the backbone. Now, what kind of bonds hold these nucleotides together? Well, here's one nucleotide and here's the other nucleotide. What holds them together is this bond right here between this sugar and this phosphate. And it's a dehydration reaction that results in a ester. So what we call these bonds are phosphodiester bonds. So this question is asking to find the hydrogen bond. And in a hydrogen bond, we're looking for a hydrogen connected to an oxygen or nitrogen, right? So in this case, we'll look at one bond at a time. So let's take a look at bond number 1 over here. We see that we have oxygen here, which is a partial negative. And we see that we have a hydrogen over here, which is a partial positive. And this will create a hydrogen bond. Over here, bond number 2, we see that we have a hydrogen and we do have a partial negative oxygen. However, this hydrogen is connected to a hydrocarbon, another carbon. So this bond is nonpolar. Since it's nonpolar, it will not have no partial charge and it will be unable to interact in a hydrogen bond. So 2 will not count. 3 is a bond between oxygen and hydrogen. This is in fact an actual bond and it's called a polar covalent bond, and that is not a hydrogen bond. Finally, 4 is a hydrogen to another hydrogen, which is not a criteria for hydrogen bonding, and that will not count as well. So in this case, we only have one hydrogen bond, which is bond number 1. So our answer in this case is B. So this question is asking about a protein and its side chains. So we can take a look at the protein on the right. We can see that if you look at the NCC structure, that we have 2 amino acids and the peptide bond is over here. So we'll cut that in half. And its respective side chains are on the center carbon. So right here and right here. And we can see that the side chains are methyl groups, which are CH3s in this case. And these are considered nonpolar, and both will be hydrophobic. So our answer choice in this case is E. So number 7 is asking that a specially prepared batch of heavy hydrogen peroxide has a mass of 38 grams per mole. Normal hydrogen peroxide has a mass of 34 grams per mole. The mass of heavy hydrogen peroxide makes most sense if it is made of. So in this case, we're gonna take each hydrogen, each oxygen, and see which heavy atom combo comes out to be 38 grams per mole. So originally, if we write out H2O2, one atom of hydrogen weighs 1 gram. And so in this case, since we have 2, we'll have 2. And 1 atom of oxygen weighs 16. So in this case, we have 2, so 32. And this is where they get a total of a normal 34 grams per mole. However, in our heavy, we have H2O2, and that totals up to be 38 grams per mole. So let's go one at a time, and we'll determine which combination equals 38 grams per mole. So if we start off with a normal oxygen of 16, we'll still remain 32 over here. And we have a heavier hydrogen, since each hydrogen is going to weigh 2 now, not 1. So in this case, it will be 4. So this equals 36, which is slightly lower than 38. So A will now count. B, we have a heavy hydrogen, and this time it weighs 3. So 3 times 2 is 6. And we also have a heavy oxygen of 17. So 17 times 2 is 34. And this total comes out to be 40, which is a little too much. So B will not count as well. C, we have a normal hydrogen. So we'll take that of 1 gram each, times 2 is 2. And we have a heavy oxygen of 17. So 17 times 2 is 34. That comes back to 36, which is still not 38. So that will not count. And finally, we have a hydrogen, normal, so 2, plus a heavy oxygen. However, this time it's 18. So times 2 is 36. And that gives us what we're looking for. It gives us 38. So in this case, it looks like our answer will be D. If you take a look at E, we have a heavy H and a heavy O, and that will total up to be 40. So that will not count as well. So in this case, our answer is D. So number 8 is asking, in nucleic acids, a base pair means that. So when you hear a base pair, you should think of DNA or RNA bases. So we'll think of like adenine, cytosine, and guanine in this case. And they base pair with another complementary strand, right? So in this case, let's just say thymine, guanine to cytosine. So as you can see, this involves 2 distinct strands, right? One here and the other strand over here. And in the middle, they're connected by H bonds, right? Hydrogen bonds. So let's keep that in mind. So if we look at answer choice A, 2 nucleotides are next to each other on 1 strand of RNA or DNA. 1 strand would not be correct in this case. So A is out. B, non-covalent interactions are occurring between 2 nitrogenous bases. Well, we do have nitrogenous bases, right? And we do have a non-covalent interaction since it's a hydrogen bond. So B does sound right. So we'll circle that for now. C, 2 nitrogen bases are joined together by covalent bonds. So they're joined together by hydrogen bonds, which are actually non-covalent interactions. So C will be incorrect in that case. D, phosphate groups are able to form hydrogen bonds between 2 strands of DNA or RNA. Phosphate groups form phosphodiester bonds, and that's in the sugar phosphate backbone. So that will not be right. And glycosidic linkages join together two nitrogenous bases. Glycosidic linkages, from what we learned, are involved in carbohydrates, not really nucleic acids. So E will not be right as well. So in this case, our answer choice is B. So let's take a look at number 9. The secondary structures found in proteins are directly stabilized by. So let's look at the 4 structures of proteins. We got primary, which are held together by peptide bonds. We got secondary structure, which is held together by hydrogen bonds. However, that's between the amino group, hydrogen, and the oxygen from the carboxylic acid, right? So from the peptide backbone specifically. The tertiary group is side chain interactions. So I'm just going to write R interactions. And the quaternary structure is interactions between more than one polypeptide chain. So in this case, we're talking about secondary. So let's take a look at each answer choice. So non-covalent bonds in the polypeptide backbone. So non-covalent bonds are hydrogen bonds, right? And in the polypeptide backbone makes sense, as we said, it's between the amino and the carboxylic acid groups. So A looks good for now, so let's circle it. B, peptide bonds between N and C atoms. Peptide bonds are in the primary structure, not the secondary, so we can take that out. Answer choice C says hydrogen bonds between polar side chains. So hydrogen bonds between polar side chains hold together tertiary structure, right? Because that's what involves side chains. So we can take that out as well. D, hydrophobic interactions between carboxyl and amino groups. So hydrophobic interactions occur in the tertiary structure once again, and it would be hard to get a hydrophobic interaction between a carbonyl and amino group since those are polar. So D would not count. And finally, E, covalent bonds between cysteine side chains. So this is referring to like disulfide bonds, and those are also found in tertiary structure, not secondary. So in this case, our answer choice is A. So number 11 is asking what is wrong with this diagram of a ribonucleoside. So let's define a nucleoside. Nucleoside only has a sugar and a nitrogenous base, no phosphate, right? So we do see a sugar here, and we do see a nitrogenous base attached to it, all right? So let's go through each answer choice. The 2' carbon should not have an OH on it. Well, let's label the carbon. So we have our 1', 2', 3', and 4'. Well, our 2' carbon does have an OH on it, and it's saying we shouldn't. Well, if it didn't, then it would be considered deoxyribose. But since our question asks ribonucleoside, we are okay. So A is not correct. 11B is asking if it is missing the 5' carbon. Well, we do see we only have 4 carbons, and a nucleoside eventually becomes a nucleotide by having the phosphate attached to the 5' carbon over here, right? So we are missing a 5' carbon. So let's take B as correct for now. Let's move on to C. It needs at least 1 phosphate. Well, if it had a phosphate, it would be called nucleotide, not side. So we'll take that out. 11, nothing is wrong. Well, we do see that a ribose has 5 carbons and this one only has 4. So something is definitely wrong. And finally, D or E, the nitrogenous base should have 2 rings, not 1. Well, there's 2 types of bases. You got purines and you got pyrimidines. And purines are 2 rings, while pyrimidines only have 1 ring. The way I remember it is that pyrimidines sound like pyramids. Pyramids have a sharp tip, and they can cut things. So cut is my acronym: cysteine, uracil, and thymine, while the leftover ones are adenine and guanine, and those are your purines. So that's just a little acronym if you want to remember it. Pyrimidines, cut. And so you do have a single ring structures. So in this case, we are okay. So E will not be right as well. So our answer choice in this case is B. So let's look at number 12. It's saying pentane and water are both liquids at room temperature. Which of these has a greater specific heat capacity? Well, first things first, it's asking for greater specific heat capacity. So we're looking something about going from like a liquid to a gas, right? And putting in heat in between. So let's look at pentane. Pentane is a nonpolar, very hydrophobic, right? While water is very polar, and it's going to be the opposite, right? Hydrophilic. So I'm just going to write polar. So let's remember these for now. Water, so let's look up answer choice A, water, because pentane cannot form a network of hydrogen bonds like water can. Well, that sounds pretty right to me. So pentane is very nonpolar, and in that case, it won't really have many intermolecular interactions besides van der Waals, while water can do hydrogen bonds, which are really strong, right? So if they're really strong, it's gonna be harder to boil, and you're gonna need to put in more heat, right? So A sounds right for now. B, we would expect a similar specific heat capacity because both can form hydrogen bond networks. So pentane is completely hydrophobic since it's just carbon and hydrogen, so no hydrogen bonds. So that does not sound right. C, pentane, because greater strength of van der Waals. Hydrogen bonds overpower van der Waals. They're a lot stronger, so it wouldn't make sense to call van der Waals stronger. So C does not sound right. D, pentane, because as a molecule, it is much larger. Well, in this case, size, it does increase van der Waal interactions. However, hydrogen bonds are still a lot stronger, and in this case would overpower. So B or D is not right as well. Finally, E, water, which is right, but however, the description is saying because it has a lower molecular mass. Well, the better description is because it can hydrogen bond. So we'll talk about A being the answer. So 13 is saying within one water molecule and an ice cube, the interaction between an oxygen and a hydrogen is best called an. So one water molecule. So let's just draw one water molecule. So one oxygen and two hydrogens. And between this oxygen and hydrogen, we have a polar covalent bond. So let's just look for that in the answer choice. And we do see that it is A. Hydrogen bonds would be in between water molecules, but in this case, we're only being asked for one, so that wouldn't work. Non-polar covalent, we know that these are both polar covalent bonds since the electronegativity difference between hydrogen and oxygen is greater. So no for C. Ionic bonds, we don't really see any ions or any metals involved, so no D. 13 van der Waals, those are mostly weak interactions, and those are intermolecular interactions, and in this case, we're talking about intramolecular interactions, so that's why it's polar covalent bond. So let's take a look at number 14. 14 is saying, examine the molecule here of the following, the most correct description is. So let's take a look at the molecule on the right. Answer choice A is saying a short chain fatty acid with 5 carbons. So a fatty acid, we would want to see some sort of carboxylic acid group, which is a double bond O to an OH. And we don't really see that in this case. We see a ketone, right? Which is not the same thing as a fatty acid. So let's take that out. B, a 5-carbon aldehyde. So let's look for an aldehyde. An aldehyde would be a C double bond O bonded to a hydrogen. We see a C double bond O, but they're both bonded to carbon. So those are ketones right there. So no B. C, a hydrocarbon that has a carbonyl and a hydroxyl. So yeah, we do see a carbonyl right here, and we do see a hydroxyl. So that sounds right. So let's mark C for now. D, a monosaccharide in linear form. Well, a monosaccharide is a carbohydrate, which means every carbon must have a hydroxyl group, and one of the carbons must have a carbonyl, right? So let's look. If we look at it, every carbon does not have a hydroxyl. Even though one of them has a carbonyl, we can see that these 3 carbons on the left only have hydrogens. So it will not be classified as a carbohydrate. So D is not right. And E, an organic molecule with a carboxyl at one end. A ketone is a carbonyl, and so that will not work as well. So our answer choice in this case is C. So number 15 is saying we have 0.01 moles of HCl added to 1 liter of water, assuming that HCl dissociates completely into H plus and Cl minus ions. The pH will be closest to So first, let's write out the concentration of HCl in exponential form. So 0.01 will be written as, we're moving 2 decimal places, right? Can be written as 10 to the negative second, right? And then we want to use the formula pH equals negative log 10 to the concentration of hydrogen ions. And in this case, we know that we have hydrogen ions because HCl dissociates into H plus and CO minus, right? So we're not dealing with any OH here. So we can plug in our 10 to the negative second straight into here. And if we do that, we can see that our answer choice will come out by, so we cancel out these 10s, right? So log cancels out, and this negative can cancel into a positive, and eventually we get pH equals 2. The answer choice here is B. So number 16 is saying a polymer is made by joining together 4 monomers of C3H6O3 using dehydration reactions. And they're looking for the final chemical formula of the polymer. So in this case, if we look at this picture over here, we're taking 4 monomers, so 1, 2, 3, and 4, and linking them together. Dehydration reactions release waters, right? So we'll just draw in advance. We're gonna lose 3 waters, right? So let's take that into consideration. Now what I like to do is, since we have 4 monomers, we'll multiply our single monomeric formula by 4. So C3H6O3 times 4 gives us C12H24O12. Now you can see that in our answer choice, we do see answer choice D the same. However, that's a trap because we are not done yet. We got to subtract the 3 waters that we lost in our dehydration reactions, right? So what I like to do is multiply 3 into H2, which gives us 6 hydrogens. And we also use 3 oxygens, right? Because we have 3 waters. So we need to subtract our three oxygens, and we got to subtract our 6 hydrogens to get our final formula, which gives us C12H18O9. And we can see that our answer choice matches up well with answer choice C. So number 17 is asking which of the molecules shown here correspond to C5H10. So what I like to do first is, I like to look for my carbons and make sure all my molecules have 5 carbons, since those are easier to count. So let's take molecule 1. We see we have 1, 2, 3, 4, and 5. So we're good. Molecule 2 has 1, 2, 3, 4, and 5 over here. Molecule 3 has 1, 2, 3, 4, and 5. So they all have 5 carbons. So now let's move on to looking for our 10 hydrogens. So in molecule 1, we have 3 hydrogens here. We have 3 hydrogens here. We have 3 hydrogens here. And we have 1 hydrogen here, because each carbon can be bonded to 4 different atoms. So in this case, we do have a total of 10 hydrogens, which does match our formula of C5H10. So 1 works. 2, we see that we have 2 hydrogens here, 2 hydrogens here, 2 hydrogens here, 1 hydrogen here, right? Because there's 3 things, so the 1 thing would be a hydrogen, and 3 over here. Now this totals up to be 2, 4, 6, 9, and 10 as well, right? So we can see that this one matches our formula as well. Looking at 3, we see that we have 3 hydrogens here, 3 hydrogens here, 3 hydrogens here, 2 hydrogens here, and 1 hydrogen here. This is just way too many hydrogens, right? We have 3, 6, 9, 10, 11, and 12. So that's too many hydrogens, 12 hydrogens. We're looking for only 10. So our answer choices are would be 1 and 2, which is E. So number 18, the mass of all the neutrons in 1 mole of 14 carbon is. So 14 carbon means that the total weight of this isotope of carbon is 14, right? So we gotta make it 14 grams. We know that carbon is defined by protons, right? So it has 6 protons, right? If you look on the periodic table, you see boron has 5, carbon has 6, nitrogen has 7, right? In this case, we have 6 protons. And if we have 6 protons, that means the remaining of our mass would be neutrons. So 14 minus 6 is 8. So 8 grams would be neutrons. So our answer choice in this case is D. Number 19 is asking what type of isomers we can see with this molecule. So let's first look for cis-trans. So cis-trans would have something where you have a double bond, and let's just say we have H's on one side, and then you'd have another atom, let's just say X on the other side, right? So that's cis, and if we were to make that trans, it would look something like this. So X over here and X over here. So in this case, we do have this type of structure. So this is our double bond, and we can have a H here and the other H is here. So this is a cis form. But we do see that this one is possible of having cis-trans. So that is one of them. So we can take off A and we can take off E. Next thing you want to look for is structural. We definitely have structural, as you can see that these bonds can be manipulated around and put in different areas and still have the same chemical formula, right? So we can take out B. And finally, we're left with C and D, and the only difference is enantiomer. So enantiomer, you want to look for an asymmetrical carbon, right? So the way we define asymmetrical carbons is a carbon attached to 4 different atoms, right? So let's take a look at each carbon. Well, we know that these carbons over here have 2 hydrogens, so those will not count. This carbon here only has 3 things attached to it since it has a double bond, same with this one. And that leaves us with 1 more carbon right here. Now this carbon has an NH2 group, an OH group, this whole chain, and it has another hydrogen, right? So since these are 4 different things, this carbon is asymmetrical and can exist as an enantiomer. So our answer choice in this case is D. So 20 is asking when a gene is expressed. So we'll start off with drawing the central dogma. So DNA goes to RNA, which goes to a protein, right? So this is like the general path of a gene, right? The process from RNA to a protein is translation. I'm sorry for the bad handwriting, by the way. And the process for DNA to RNA is called transcription, right? So we'll just go at each question one at a time. So, or each answer choice. So A is saying DNA directly undergoes the process of translation to make a polypeptide. Well, DNA cannot just go through translation. It has to go to RNA first. So A is for sure not right. B is saying RNA sequences are used to assemble amino acids in a specific order. Well, that's true, right? RNA gives the code for to be translated into an amino acid sequence, which eventually becomes a protein, right? So A sounds right. So let's circle that. Or B sounds right, my bad. C is saying DNA gets chemically modified by removal of functional groups to become RNA. Well, we know that transcription is not like removing anything. It's more of like assigning the complementary strand, right? So removal doesn't really make sense. And functional groups, it's more of a nitrogenous basis. D is saying proteins are folded by their physical interactions with nucleic acids. Proteins are folded by interactions between like amino acids and their backbone and side chain, right? Not nucleic acids. That doesn't make sense. And E is saying RNA undergoes transcription to make a polypeptide or protein. This word we're looking for is translation, not transcription. So E is not right. In this case, our answer is B. So 21 is asking, with very few exceptions, every protein found in a cell in your body corresponds to. So A is saying structural role in keeping cells intact. Well, this is referring more to maybe like cell walls or cell membranes, which are phospholipids or even glycosidic. So this doesn't really make sense. B is saying function as a catalyst for a chemical reaction. Well, we have heard of catalysts being enzymes, However, enzymes are proteins. However, they're just very specific types of proteins. Right? We have more than just enzymes plus proteins in our body. So B is very specific. C, carbohydrate polymer with the same size and shape. So carbohydrate is a whole different macromolecule. We're talking about proteins, so C doesn't make sense. D, specific enzyme responsible for its synthesis. So again, we're talking about enzymes, which is a specific category of proteins. So not every protein can be classified in this enzyme category. So D is not right. And E, segment of DNA that determines its structure. So this sounds right? So in the slide we just talked about before, DNA determines the RNA sequence, which gets translated into the type of protein it will be. So ultimately, what you have in your DNA determines the protein structure. So E is our correct answer in this case. So 23 is asking, an atom of 33 sulfur undergoes a type of radioactive decay in which a proton decays into a neutron. Assuming that no particle leaves the nucleus, what atom results? So I drew out the periodic table here. We go phosphorus, sulfur, chlorine, and these are the atomic numbers. So they indicate how many protons each element has. So we're looking at sulfur here, and we see that it has 16 protons. And it's saying that a proton decayed into a neutron, right? So meaning that we're gonna lose one proton, and it's gonna become 15, right? However, our mass would stay the same, right? Because a proton became a neutron, and since they each weigh the same, we're just exchanging, right? So we'll make it 15, and the mass is still 33, right? So 15 protons and 33 grams. So what would that be? Well, we look at the periodic table. Atomic number 15 is phosphorus, and it'll still be 33. So our answer is B. So 24 is saying an interaction of two hydroxyl groups in a dehydration reaction could form from a glycosidic bond or a peptide bond. So let's talk about glycosidic bonds. So they're between two carbohydrates, right? Every carbohydrate we know has OH groups on it, right? So we have an OH on this side, and let's say we have another OH on this side. So these are two monomers, and they combine to dehydrate into a polymer with a glycosidic linkage in between, and they release a water, right? So this is a valid dehydration reaction. Peptide bonds are between amino acids. So if you take two amino acid structures, so N, C, C, and on this side we have a carboxylic acid group, and over here we have a N, C, C, right? And this nitrogen has two H's, right? Not an OH. However, even though it's a dehydration reaction, it is not an interaction between two hydroxyl groups, right? It's just between an OH and an H. So this is a very specific question, and the answer would only be glycosidic bond. In this case, our answer is A. So our last question of the day, we have a number of asymmetric carbons in this molecule. So let's take a look. We know that, as talked about earlier, asymmetric carbons come from a carbon attached to 4 different atoms, right? So we'll go through it one carbon at a time. So let's look at this carbon over here. We have 2 hydrogens, right? So automatically that disqualifies this as an asymmetric carbon, so no. If you look at carbon 2, we have a hydroxyl group, we have a hydrogen, we have this carbon chain, and we also have this carbon chain, right? So that's 4 different things. And that makes this an asymmetrical carbon. So I'll put a check mark. Let's look at the third carbon. We see that we have an OH, we have an H, we have this chain right here, and we have this chain right here. That's 4 different things, so this one counts as well, right? This carbon right here only has 3 things attached to it. Remember the rule was 4, so no. And finally, this carbon over here has 2 hydrogens, so that's not 4 different things, so that will not count as well. So that leaves us with 2 asymmetrical carbons, which is answer choice E. And that is all. So sorry for going really fast, but go ahead and pause the video and try it yourself before looking at the answers. And I apologize for the bad handwriting, but good luck on your midterm tomorrow. Thanks.